Hey guys, so the first thing I'm going to do is write that these two strands are anti-parallel to each other, which just means if one end is 5', then the other has to be a 3'. So for the first part, we're going to look at how do we synthesize a strand continuously. So this would be forming our leading strand. But before we do that, we need to actually separate our DNA strands, which we are going to do with an enzyme known as helicase. Right? So what helicase does is as it moves along, it simply separates the two DNA strands from each other. So it unwinds the double helix. In order to keep the strands apart, I'm going to draw our SSBPs, which is short for our single strand binding proteins. So I'm going to color them in, and they're on both strands. And their job is to simply keep the strands apart, prevent them from recoiling. So now I'm drawing on an RNA primer. So because one end was 3', that means this over here is going to be 5'. And our RNA primer functions as a starting point for the enzyme DNA polymerase 3, which I'm going to draw on. Because without it, DNA polymerase 3 doesn't know where to go. So now that I have my DNA polymerase, we can begin our DNA replication. So I'm just going to label it here. So we have DNA polymerase 3. So we had that on the end of the primer, it was a 5' end, which means this end over here has to be a 3', which is important because all around the DNA we have free-floating nucleotide bases. Right? We have a G, a C, an A, a T. And in order for them to get added, it can only be added to the 3' direction, which means DNA polymerase has to be moving towards the fork. Okay, so the DNA polymerase 3 moves towards the fork, and as it does that, our bases get added to the 3' end continuously. Okay, so that's what I'm drawing on here. Okay, so that they're all getting added continuously. If we had a 5', which means this end over here has to be a 3'. Because DNA polymerase 3 built this strand continuously, we have a specific name for this strand, and we call it the leading strand. So once again, that's just when we have our DNA is being built from 5' to 3' continuously. Now let's look at our opposite end. So see this primer over here? Well, now what we're going to do is actually see how was it created. And that was because of an enzyme called RNA primase. And its job was to simply go through the strand and create these RNA primers. And right away you can tell we have multiple primers here, where the leading strand only had one. So once again, the name of this enzyme is RNA primase, responsible for creating the primers. And once again, the primer's job is to act as a starting point for our enzyme DNA polymerase 3, which I'm going to draw on again. And it has the same structure as the DNA polymerase 3 I drew before. It's the same enzyme. Okay, so it's DNA polymerase 3. So same thing. This end has to be, because it was a 3 over there, it has to be a 5' to start with, which means this end over here has to be a 3'. So my question then becomes, this DNA polymerase 3, is it going to move towards the fork or away from the fork? And because it can only add nucleotides to the 3' end, it has to be moving away from the fork. That's the only way it'll be able to add it to the 3' end, if it's moving away from the fork. So what it does is, as it's moving away from the fork, it's adding DNA nucleotides after the primer. So that's what I'm going to draw here. After every primer, DNA nucleotides are added. And this end over here is just a 3'. So now I have these 3 fragments. Over there it was continuous, here not so much. And these fragments actually have a name. And I'm going to label this one, and it is called the Okazaki fragments. And that's just when we have an RNA primer and a fragment of DNA. So in this case, I've got 3 Okazaki fragments drawn. So the thing is, though, I still have these RNA primers, and this is DNA replication. I only want DNA. So I'm going to introduce another enzyme, which I'm going to draw in as a star. So I'm going to draw my star, color it in, and this star is going to represent the enzyme DNA polymerase number 1. And the job of this DNA polymerase is to move along the strand, and every time it sees an RNA primer, it changes it to a DNA nucleotides. Okay, so it replaces every RNA nucleotide with a DNA nucleotide. Okay, so as it moves forward, it's going to do the same thing here. We're going to get rid of our RNA, and it's now going to be DNA. All right, we're almost there. So now we got rid of the primers, which is great, but we still have fragments. So we're going to introduce this last enzyme, which I'm going to draw in as a heart. So I'll just draw a heart over here. And what this heart is going to represent is the enzyme, and there's not really much space here, but the enzyme DNA ligase. And all it does is just move along the strand and simply connect the fragments together.